What is carcinogenesis? Carcinogenesis is the process through which normal cells transform into malignant cells, resulting in cancer formation and progression. Factors that contribute to cancer formation are genetics, unhealthy lifestyles, exposure to certain chemicals, and chronic inflammation. What is molecular and cellular signaling? Molecular signaling can be defined as the interaction between molecules inside the cells that leads to regulation of the normal cell function. This includes proteins, receptors, and also components of the cellular pathway. Meanwhile, cellular signaling can be defined as the coordination of numerous cells throughout tissues and organs. It includes cell-to-cell communication, as well as interactions between various cell types within a tissue. Before we start on how cancer develops, let's see on how the normal cell develops first. Generally, cell cycle divides into two phases, which are interphase and mitosis. During interphase, the cell grows and makes a copy of its DNA, while in mitotic phase, the cell separates its DNA into two sets and divides its cytoplasm, forming two new cells. Interphase contains G1 phase, S phase, and G2 phase. During G1 phase, also called the first gap phase, the cell grows physically larger, copies organelles, and makes the molecular building blocks it will need in later steps. In S phase, the cell synthesizes a complete copy of the DNA in its nucleus. It also duplicates a centrosome, which helps separate DNA during M phase. During second gap phase, the cells grows more, makes protein and organelles, and begins to reorganize its contents in preparation for mitosis. G2 phases end when mitosis begins. Let's move on to mitosis. During the mitotic or M phase, the cell divides its copied DNA and cytoplasm to make two new cells. M phase involves two distinct division-related processes, mitosis and cytokinesis.
Overall, the cell cycle is a complex and meticulous process to ensure the survival of a cell. In order to ensure that the cell divides accordingly, the cells themselves have their own internal quality control mechanism, which are also known as cell cycle checkpoints. There are three checkpoints that are most understood, which are G1 checkpoint located in between G1 and S phase, also known as the restriction point. Next is G2 checkpoint, which is located in between G2 and M. And last but not least, the spindle checkpoint, which is located in the M phase between metaphase and anaphase. Let's take a look at the G1 checkpoint. This checkpoint checks whether the internal and external environment of the cell is suitable or not to carry on with the division. Any errors or abnormalities in the DNA will be detected, and the cell cycle will be stopped temporarily until they are repaired. If the errors are irreparable, the cells will go for programmed cell death or apoptosis. Two main components involved in the cell cycle regulatory mechanism are cyclin-dependent kinases, CDK, and cyclins. CDKs are a type of enzyme that is responsible to phosphorylate other proteins in the cell to activate or inactivate them. Cyclins is a type of protein that will bind to CDKs to activate it and form cyclin-CDK complex upon receiving signals from signaling molecule. Different types of cyclins are produced in different concentrations in according to checkpoints respectively. The actions of the cyclin-CDK complex will trigger a phosphorylation cascade, which induces cellular response, and are also the green light for the cell to proceed with the cell cycle.
Negative cell cycle regulation. What is negative regulators? Negative regulators play a vital role to stop the cell cycle progression by preventing it from passing the checkpoints. The best understood negative regulatory molecules are retinoblastoma protein, RB, PT53, and P21, which is also known as the tumor suppressor protein. RB, P53, and P21 primarily act at the G1 checkpoint, assessing DNA integrity and determining whether the cell may advance to the S phase or not. If damaged DNA is recognized, P53 stops the cell cycle and recruits enzymes to repair the DNA. If the DNA cannot be repaired, P53 can induce apoptosis or cell death to avoid chromosomal duplication, which can lead to the cancer formation. Then, the production of P21 is stimulated by the rising of P53 levels. P21 enforces the halt in the cycle dictated by P53 by binding to and inhibiting the activity of the CDK cyclin complexes. The stress condition caused by the accumulation of P53 and P21 makes it less likely the cell will continue to the S phase. Mutated P53 does not arrest the cell cycle. The damaged cell continues to divide, which may result in cancer formation.
What is apoptosis? Apoptosis is a programmed cell death in which the cell activates a signaling cascade that leads to cell death without triggering an inflammatory response. Why do cells undergo apoptosis? To eliminate the abnormal cells such as stressed DNA damage and hypoxia, mechanism for preventing cancer and natural homeostatic mechanism to maintain cell populations in tissues. This is the core mechanism of both extrinsic and intrinsic pathways in the normal regulation of cell apoptosis. The extrinsic pathway happens when the cell receives stimuli from outside by TNF receptor and instructs the cell to plan cell death. The outside signal molecules bind to Fas receptors and trail receptors forming ligands. FAD responds to this cellular change and binds to the ligand. Two more inactive proteins called procaspase 8 and procaspase 10 interact with FAD, forming a death-inducing signaling complex, DISC.
DISC activates caspase 8 and caspase 10 and spreads in the cytoplasm. Then activates the executioner, caspase 3 and caspase 7, and begins the cleavage of proteins and the cytoskeleton leading to cell death. In other pathways, from procaspase 8, the activated caspases cleave off other inactive molecules known as BID, BH3 interacting domain death antagonist to convert into TBID and become activated. TBID then transported to the mitochondria, stimulates the activation of BAX and BAK molecules entering the intrinsic pathway. In intrinsic pathways, internal cellular stimuli such as DNA damage, hypoxia, growth factor withdrawal stress, or chemotherapeutic agents alter the cell's capacity to keep its function and lead to apoptosis.
BAX is regulated by P53 and triggers the pro-apoptotic BH3-only protein upon stimuli. The permeability of mitochondria outer membranes known as MOMP is altered, releasing cytochrome C into the cytoplasm, which functions as signaling molecules. Then, cytochrome C binds activates APAF1 and procaspase 9, forming an apoptosome, a protein complex that initiates the phase of cellular degeneration. Procaspase 9 converts into caspase 9 and then activates caspases 3 and caspases 7. The degradation of cellular components and degrade the DNA of the cell initiated, leading to cell death. So what is the relationship between carcinogenesis and apoptosis? Mentioning DNA damage as an example, cancer cells will continue their proliferation since tumor suppressor protein P53 loses its function in controlling cell replication and escapes the apoptosis process. BH3-only protein, which encodes pro-apoptotic protein, also cannot be activated. Other pro-apoptotic proteins become inactivated, and the caspases inactivate, leading to dysregulation of apoptosis. The cells continue dividing without being programmed to cell death.
To summarize, the complicated world of molecular and cellular signaling is critical in the complex process of carcinogenesis, or the formation and development of cancer. The mixture of abnormal signaling pathways, genetic abnormalities, and environmental effects can upset the delicate balance of cellular homeostasis, sending normal cells down a road of uncontrolled proliferation, apoptosis evasion, prolonged angiogenesis, and tissue invasion. Cells can develop cancer-like characteristics as a result of dysregulated interactions between growth factors, receptors, kinases, and downstream effectors, ultimately leading to tumor development. Here are our references. Alright, I think that's all from us. Hopefully this video bring benefit to you.